us today, Isabelle Fudal. So welcome, Isabelle. You're in Paris. So that's like we spoke a little bit before. There's 6 time zone in Canada, so the very early people in West Coast are, yeah, early in the morning, and for you it's almost dinner time. So I welcome you and I thank you for being here. You are a senior researcher at the French National Institute for Agriculture, Food and Environment. It's well known as INRAE. And you are in the unit that's actually dealing with fungal biology and risk management in agriculture. If I'm correct, it's called BioG.
Biogène, but I will be presenting the institute.
Okay, excellent. So that's very good. And your background is very much plant pathology all along. If I'm correct, you've done an engineer degree from the very well-known AgroParisTech, or formerly INAPG school in Paris. And then all along your PhD and postdoc, and until now was about plant and fungal interaction.
Sorry, I don't hear you anymore. And
Oh, suddenly
yeah, but okay.
mute.
No, I
I'm
don't know.
sorry. I don't know. I didn't
I
touch
don't know
anything.
if I cut something
And I
off.
got muted, so I don't know what you
That's
missed.
okay. No, that's
I
great.
was, yeah, I was telling that you have all along a background on plant pathology. I don't know if yeah, and
Yes.
From your engineer degree, right, at AgroParisTech, formerly known as INAPG, Institut d'Agriculture in Paris-Grignon, and then your PhD and postdoc were all about effectors, plant and fungal interactions, and defense mechanisms. So with that, and with no further ado, please, you may begin.
Yeah. Thanks a lot, Isabelle, for this invitation to present in this network of fungal people interested by fungi. So it's a great pleasure today to present you part of the work that I'm developing in my group on Leptosphaeria maculans. It's a fungal pathogen of rapeseed or oilseed rape. And as you said, I'm a senior scientist at INRAE, and I'm working at the BioG Institute located close to Paris. So the BioG Institute was previously located in the west of Paris and recently moved to the south to a new area of in the new campus of AgroParisTech in the University Paris-Saclay. So new area, new buildings, and new facilities. So BioG is entirely dedicated to the study of fungal pathogens of crops. And in this institute, we develop interdisciplinary approaches at different scales, from the fields to the gene and genomes and back to the fields. So we develop different strategies to do that, molecular biology or biochemistry, cell biology, but also genetics, genomics, evolution, epidemiology, and even modeling. The different fungal models which are studied at BioG are main pathogen of wheat, rapeseed, Arabidopsis and cabbage and grape. So on wheat, people study pathogen responsible of different rust, the Puccinia species. Also, Zymo-Septoria tritici, responsible of septorial leaf blotch. Botrytis cinerea, which is responsible of disease on many different crops. It's responsible of the gray mold, and it's a pathogen also of grape. And Coletotrichum gentianum, pathogen of cabbage and Arabidopsis. And finally, Leptosphaeria maculans, the fungus I'm studying in my team, a pathogen of rapeseed. So, and on all these models, we want to find ways to fight these different pathogens, either using resistances of the plant, the ability of the plant to resist to the infection, or the use of fungicide or biocontrol methods to fight these different diseases. So to start, I wanted to give you some information or reminders about the way pathogen will infect a plant. So the pathogen, when they infect the plant, they secrete a set of molecules called effectors, which can correspond to small proteins, to secondary metabolites, or to small RNA. And all these molecules are able to manipulate the plant and this way to facilitate infection. But in some cases, these effectors are recognized by the plant machinery. And in this case, there is an induction of defense reaction and infection is stopped. This recognition is done by resistance proteins, which can be cytoplasmic or recognized effectors which are apoplastic. So we have two main categories of resistance protein according to their localization. And in all these cases, these effectors, which are recognized by the plant, are called avirulence effectors, because in this case, there is no infection anymore. So the use of specific resistant gene of the plant, it's a very efficient way to control the disease. However, if you use at a large scale only one resistant gene in the fields, then you will have the pathogen will adapt quite rapidly and escape this recognition and will be able to infect the plant. Here you have the example of a resistant gene which was introduced in rapeseed like 30 years ago, and in a few years of use, in almost 3 years, the pathogen became mainly virulent towards these resistant genes. And in fact, the corresponding avirulence gene was lost in the populations. So that means that we need to improve the way the plant resistance are used and to increase their durability. And for that, we need to know more about avirulence effectors, their role, and the way they evolve, their evolutionary dynamics in the populations. And we do that in close collaboration, both with academic and socioeconomic partners. So here I will present you the fungal model we study, so Leptosphaeria maculans, responsible of stem canker of rapeseed. It displays a quite complex infection cycle. The fungus first stay on stem debris. In France, it's during the summer, but really depends on the way you grow rapeseed. Either you have winter rapeseed or spring rapeseed. But in France and in Europe, the pathogen developed on the stem debris and will do its sexual reproduction. It disseminates them sexual spores, which are called ascospores, which are disseminated by wind and will infect young leaves and cotyledons of rapeseed. You will see in autumn some necrosis on the leaves, but at that stage, you can have secondary, asexual reproduction and secondary infection. But then, what is particular to this pathogen is that it will stay into the plant after this stage of infection. And it stays into the plant without causing any symptom during several months. And then it migrates to the same stem basis at spring. And here it causes the stem necrosis. So the main damaging symptoms on the plant. So the main way to control this disease is the genetic control using specific resistant genes called RLM genes in this model. And when you have a resistant plant, then the fungus is recognized very early when it infects the leaves and infection is stopped. What is recognized are avirulence gene, and in this model, these avirulence genes are called AVR-LM genes. This specific resistance is often combined with quantitative resistance that will limit the colonization of the stem and the development of the stem necrosis. In this model, we have been able to clone 12 avirulence genes. It's quite a lot for a fungus. Most of them were cloned by our team and also some by colleagues from Canada and others from Australia working on this system. And we were able to determine some common characteristics for these avirulence genes. So most of them are upregulated during the biotrophic stages of infection. So we have a lot of data on the expression kinetics all along the infection cycle of Leptosphaeria maculans. Here you can see condition in vitro growth conditions, infection of cotyledons, of petiole and stems, and you can see a very concerted, well-concerted expression of this avirulence gene at the different stages of infection and an upregulation during the biotrophic stages of infection. The other point is that these genes are located in particular regions of the genomes, which are repeat-rich regions. And this has consequences on the way these genes are evolving when a new resistance is used in the field, because they are able to evolve very rapidly, particularly during the sexual reproduction that occurs every year through deletion of these repeat-rich regions or accumulation of mutations, because in ascomycete there is a mechanism called RIP, for repeat-induced point mutations, which is active before meiosis, that will recognize the elements which are repeated in the genome and will mutate these elements. So this is a very efficient mechanism that inactivates the transposable elements. But since these genes are located in the middle of transposable elements, they are also mutated and inactivated through this mechanism. This avirulence gene encodes small secreted proteins, which are often cysteine-rich, without homologs in the databases. They are often species-specific or genotype-specific. And finally, at least in Leptosphaeria maculans, in this interaction between Leptosphaeria maculans and Rhapside, we see quite complex interaction. We do not have always just one avirulence gene recognized by one resistant gene, but we have some more complex situation with one avirulence protein recognized by two different resistant proteins, or two avirulence proteins necessary to induce recognition, or also one case of effectors that suppress recognition of two others by their resistant genes. So I will explain in more details this complex situation a bit later. So just wanted to mention first a few of the projects we develop on this model. We want to know what is the biological function of these avirulence effectors, what are the cellular processes and plant proteins which are targeted by these effectors, and in which compartment they are acting. We try to understand this complex interaction between avirulence effector and their resistance protein, and to determine the mechanism by which the pathogen escape recognition. And we are also interested by the way this concerted expression of effectors, avirulence effector genes occurs. If we have some, what is the role of the chromatin structure, the transcription factor, and the plant and environmental signal on this regulation. So today I will focus on two of these topics, particularly interaction between avirulence effector and resistance protein, and a few, and also on the project on the regulation of these effectors. So before continuing, I just wanted to know if you had some question about this introduction, and I will stop at different moments in this presentation to have some questions. Okay. Yes.
Is that me?
Yes, it's you, Chris.
I guess, yeah, just a quick question is, you mentioned the RIP mechanism. I was wondering if you know how prevalent it is in maculans.
How, sorry, I didn't get the end of the question.
How prevalent, how commonly do you see it?
We see it at each sexual reproduction stage. It occurs, it always occurs if there is something to inactivate.
Right.
We know because all the transposable elements which were present in the Leptosphaeria maculan genome are already inactivated. So we can just see that they are not active anymore, and that there are a lot of mutations and that lead to the appearance of stop codons. But when we see that even the gene which are in the middle of transposable elements, even if they are not inactivated, we see that they are their evolution is really linked to RIP.
Okay.
There's always some RIP.
Thank you.
Vega, you may go.
Hi. Just I have a question about the last previous slide that you mentioned about the gene is located in a rich repeat. So I didn't understand what can be the effect of if the gene is located in the gene in a rich repeat. So what can be effect of that? It means that it can inactivate the gene. I didn't understand. Can you explain a little bit about that?
So I'm not totally sure about the question, but I will try to answer. What is the reason why they are located in
Yes,
these regions?
not the reason, what's the
or
effect
how it
of that?
occurred?
Yes.
What we know, it's quite something quite common in fungal pathogen. It was also seen for oilseed genomes that we have some effectors which are located in repeat-rich regions while others are located in gene-rich regions. And what we see is that these effectors are evolving more rapidly. So that means that's a way for that's more dynamic regions of the genome and a way to adapt quite rapidly to the plant defense mechanisms, to plant targets, which are evolving also. So we see that the genes which are located in the gene-rich regions are almost not polymorphic in the population, while these genes located in repeat-rich regions are evolving more rapidly. So it's really a way to, for the pathogen to evolve more rapidly.
Mutation.
And you will see in the last part of my talk that it's a way also to control very efficiently the expression of all these sets of genes through the chromatin structure.
Okay, thank you. Okay, thank you so much.
Okay, thank you. If there are no other questions, I will continue. So, and starting with the investigation of some complex interaction between avirulence effector gene and their resistant genes. So I will present you two examples and two families we studied. The first one is the avirulence gene avirulm10. So as I previously mentioned, avirulm10 in fact correspond to two genes, which are two neighbor genes separated by an intergenic region. And the two genes are necessary to induce recognition by the resistance protein RLM10. This work was done by Johan Petit during his PhD, so he could demonstrate that in fact the two genes were necessary and the inactivation of only one of the two genes allowed to escape recognition by RLM10. avirulm10A and 10B, the two corresponding proteins, physically interact. And we found that there were many homologs of these two genes in plant pathogenic fungal genomes, including several copies of this gene in Leptosphaeria maculans itself. So Nasera Talbi, during her PhD, characterized this family of effectors in Leptosphaeria maculans. So we have 5 effector pairs in the genomes that display a quite high level of homology, or high level of homology for effectors. It's not so high, but for effectors, it's already quite a lot. The corresponding proteins share the same size, same number of cysteines. They have predicted signal peptide. But the genes, some of them are located in repeat-rich regions and others located in gene-rich regions. When we look at their expression, the ones which are located in repeat-rich regions are expressed at all the biotrophic stages of infection on cotyledons, petal, and stem, while the ones which are located in gene-rich regions are only expressed during the late stages of infection. And we always see a very well co-regulation of the two genes within each couple of effectors with the two expression patterns that I mentioned. So Nasera tested the interaction between the corresponding proteins. As I already mentioned, avirulm10A and 10B are physically interacting, and we could also find a physical interaction for one of the effector pair. And that was not the case for the other. We tested this by co-immunoprecipitation and Fred Flim. Then what we did, we look for homologs of these different effector pairs in different fungal genomes, and we found 71 EVRLM10A homologs in plant pathogenic fungi from the Tidomycete and Sordiomycete families in 31 species, in fact. And for EVRLM10B, we could not find so many homologs. So this was a collaboration with colleagues Martin Rapp and Lieke Fokkens working on Fusarium, because they also look at potential homologs of EVRLM10A in Fusarium. And what we found, in fact, we found we could construct a phylogenetic tree with 3 different clades. And here with colored square, you can see if there are, as neighbor genes, some neighbor genes that encode for potential effectors and that could form an effector pair with this EVRLM10A homologs. So in 2 of the clades, we have a potential partner for this effector. So the EVRLM10B homologs are all located in the same part of the phylogenetic tree. And we could, so as I already said, EVRLM10A homologs are associated with different effector families, according to the colors you can see here. And one of the EVRLM10A homologs is a well-known effector of Fusarium oxysporum, which also has a neighbor gene, a neighbor effector gene. It's 6.5, which is with AVR2, necessary to induce recognition by a resistant gene of tomato. So it's exactly the same situation as EVRLM10A and 10B. The 2 genes are necessary to induce recognition by I2. The corresponding proteins are also physically interacting. And recently, Frank Take and his team found that 6.5 is in fact transporting AVR2 from cell to cell via the plasmodesmata of the plants. So we made the hypothesis that perhaps EVRLM10A 6.5 are conserved cooperating effectors that facilitate the transport of other effectors from cell to cell in plant pathogenic fungi. So we tested that hypothesis in collaboration with Frank Take and Mila Blekemolen. And I will not detail all the experiments we did. You can have a look at the recent publication we made, but I wanted to mention that some of the EVRLM10A homologs locate at the plasmodesmata and are able to transport their effector partner from cell to cell. And they are also able to replace 6.5 in its function of transport of AVR2 from cell to cell. So now we think that probably we have a conserved mechanism of pathogenicity mechanism in many different fungal pathogens. So now I will switch to, well, before switching to this second story, I wanted to know if you had some question on this part of the story. And then I will move on. No? If not, I will continue. So the second story of complex interaction is this story of EVRLM47, which is recognized by 2 resistant genes and itself able to suppress the recognition of other effectors by their resistant protein. So RLM4, is a resistant gene which is present in rapeseed cultivars since the early 70s in Europe. So in Europe, the population, the Leptosphaeria maculans population, are mainly virulent, so able to attack these cultivars. And when we look at the polymorphism between avirulent and virulent isolate, we found only 1 mutation that led to 1 change of amino acid. And since we were able to determine also the structure of EVRLM47, we could locate this polymorphic amino acid. It is located on the surface of the protein, and just this change allowed to escape recognition by RLM4. The situation with RLM7 is a bit more complicated because RLM7 was introduced in commercial varieties in Europe about 20 years ago. And so we were lucky to be able to follow the first event of switch to virulence. And we could see that there were many, many different events of switch to virulence, complete or partial deletion of the gene, mutation due to RIP, point mutation, or downregulation of the expression of the gene. So, and the point mutations, when we look at the polymorphic amino acid, again, we had some amino acid located close to the one responsible for the switch to virulence towards RLM4. And then also we identified another region involved in the recognition by RLM7. But what we also noticed when the population became virulent toward RLM7, we see appear some isolates that were avirulent toward RLM3. In fact, RLM3 is used in the field since also the 70s. And we thought that since in the population we could see only isolates that were virulent toward RLM3, we thought that the RLM3 gene was lost. But in fact, it's always present in the population, but its presence is masked by RLM47. So we try to determine why and how, what was the mechanism involved in this suppression of recognition. And we know now that avirulence-3 and avirulence-59 are always present in natural isolates of Leptosphaeria maculans. They are probably quite important for the pathogen. When we try to inactivate avirulence-3, it's quite difficult. And we see a major effect on the pathogenicity. We could not see any physical interaction between the three effectors, at least by yeast hybrid. And when avirulence-47 is present, there is still an expression of avirulence-3 and avirulence-59. So avirulence-47 is not suppressing the expression of the other gene. So we decided to determine the structure of avirulence-3 and avirulence-59 in order to try to understand this mechanism of suppression of recognition by avirulence-47. And surprisingly, we found that the three effectors belong to the same structural family. It was not obvious because at the protein level, there is no homology or a very, very low level of homology. So it's surprising, but in fact, we have the same structure. They belong to the same family. And we call this family LARS for Leptosphaeria, avirulence, and suppressing. On the plant side, the corresponding resistant protein were also recently cloned, for part of them by some Canadian colleagues. RLM9, RLM7, RLM4 are in fact allelic, and they encode for receptor-like kinase, so located at the plant membrane with a WAC domain. So on one side, we have a structural family. On the other side, we have some allelic genes. So we can make now some hypotheses about the mechanism that occurs, even if we still do not have the end of the story. There is possibly a competition for a common target, but when we try to identify the targets of these different effectors, we could not find any target in common. Or avirulence-47 perhaps interact with RLM-3 and RLM-9 and impaired the recognition of the effectors. And we can imagine the possibility to develop some resistance protein that could be able to recognize several members of the LARS family and to increase the spectrum of recognition of this resistant protein. I wanted to mention that we have additional member of this LARS family, Leptosphaeria maculans, one additional avirulence protein, avirulence-S-Lep2. The corresponding resistant protein, in fact, 3 of them are currently used in the field in France. So most of the resistant proteins which are currently used target the same structural family. And we also found members of the LARS family in over plant pathogenic fungi, Notidomycetes and Sordiomycetes. So now we have some new questions. So do effector genes belong, in fact, to a limited set of structural family? That's something that starts to be seen for different, when you look at the effectors of different fungal pathogens, you see that, in fact, you have conserved structural families, even if we do not have homology at the protein level. We would like to identify some resistances that will recognize new structural family and not target always the same family. And we would like to know also if when we use one resistant gene targeting one effector family, does it exert a selection pressure on the whole family or just on one effector? And finally, we would like to be able to develop broad spectrum resistances against several members of one effector family from one pathogen or even from several pathogens. So we started testing that possibility with a proof of concept. So we identified members of the last family in different fungal pathogens. Here, Cladosporium fulvum, Fusarium oxysporum. Some homologs of avirulence-free, so ECP11 and FO63. We transform Leptosphaeria maculans with the corresponding genes, and when we infected rapeseed with carrying RLM3, we could see a resistance. So the members of the last family from different pathogens are able to induce recognition by the same resistant gene. So that's just a proof of concept for the moment, but we would like to increase this, to continue to study that possibility. Okay, so yeah, finish with this first part of my talk. So if you have some question on these complex interactions, so questions are welcome. So,
hi, can you hear me?
yes.
Hi, I'm just curious. So is one of the working hypotheses, could you go back a couple of slides actually, the exact
Okay.
sorry, it's just I forget which AVRLMs are which. Just one more, I think.
One more, is this one?
Yeah, here, yeah.
Okay.
So is a working hypothesis then, just so that it's clear to me, that LM4 is binding to RLM3 and RLM9 and 5, but in a way that does not activate immunity?
Yeah,
and thereby
we could
suppresses the
imagine
phenotype
that.
associated with AVR?
Yeah, of course, it works only if it doesn't induce recognition. But we don't know for the moment how this could happen. And we don't know if there is indeed a direct interaction or not. The colleagues who are working on the resistance protein, they tested for the moment, I guess, the interaction between RLM9 and AVRLM59, and they couldn't see interaction, at least using yeast to hybrid, but it's quite complicated to work with this
yeah,
protein.
especially because you'd be working with a transmembrane protein, and presumably
it's a
you'd expect them.
protein, so they also tried just to produce the LRR part, the part which is
the extracellular domain?
Extracellular,
Yeah.
the extracellular domain, and just working with the extracellular domain, they could not see any direct interaction. That's not so easy to work with proteins with transmembrane domains. So
No, of course. Yeah, because like in theory you could do like a mutagenesis analysis, but it really only works if you have an interaction to work with.
Yeah, so we still don't know if there is a direct interaction or not. And in fact, also this new category of resistance protein, the WAC family, is not so well known. We don't know the way they interact with the effectors, because they are supposed also to recognize in plants some damaged protein, the protein which are when the pathogen enter into the plant and degrade the membrane, the cell wall of the plant, it's supposed to recognize some elements of this degradation. So
yeah, I think I've also seen some that are also just recognizing MAMPs or PAMPs or something. So presumably there's a lot more work to do there, and it may vary from case to case. Just one other question, I'm sorry to monopolize here. have you ever explored anything like N-Benth methods for looking at protein interactions or protein-protein interactions in any of these cases?
Protein-protein interaction directly, you mean by producing the proteins, or?
Well, like, can you do BIFC or something like that?
Well, BIFC, we have not tested, at least for this case. We have tested for some targets we identified, but not in this case.
Okay.
We tried some some immunoprecipitation, but we didn't find the resistant protein. So
Okay.
for the moment, yeah.
Okay. Thank you very much for your answers.
Thank you. There was another question, I guess.
Yeah, I have a question for you.
yeah.
Nice talk so far. Yeah. So the question is, so in the last slide you talk about the RLM RM3, I think. Yeah. Interacting with the multiple pathogens. So we all the examples so far from maculans, they'll show a direct interaction with AVRs and R gene. So appears to be. Is it so? What do you think is happening here with the Cladosporium as well as Fusarium? Are do you think they're direct or indirect?
Well, I guess it's, well, we still don't know, but for the moment, no direct interaction were found. So, but that doesn't mean that it doesn't exist. But for the moment, we could not see any direct interaction. And I wanted to mention here that, of course, Cladosporium and Fusarium, the one we use, are not pathogenic of rapeseed. So it's just a proof of concept.
Right.
So we need to find also effectors which are conserved between pathogen of rapeseed.
So it'd be interesting to see if there's any, probably a common Gardaí, for example.
Yeah, that's also a possibility, yes.
Right. Yeah. And I'm not sure if finished the talk or not, but Lisa, one question from your first part of your presentation. Interesting, you have the life cycle where you have initially you have the infection on the leaf, and then somehow later part of the infection cycle, it becomes endophyte.
Yes.
Any thoughts on what the hell's going on there?
We really don't know what happens at this stage, and it's quite complicated to know because the quantity of pathogen fungus at this stage is very low. So we have some data from RNA-seq data, but of what happened in the field. But during this period before spring, we had so few, so low number of fungal reads that we could not determine what the fungus was doing at this stage. So we still don't know what happens.
Yeah, very exciting. Yeah.
There's a comment in the chat from Bradley. It said, Gopal's question, I'm half remembering a paper from a few years ago where an extracellular fungal effector cleaved the plant protein, and that cleavage product ultimately activated a transmembrane receptor from the extracellular portion.
Wow.
Yeah, I just bring it up as an example of like you could have an indirect recognition mechanism driving this. So it's very exciting. Move forward.
That's a CF2 or AVR2 thing interaction, if I remember right.
Or CF2 AVR2?
I remember. I think that's what I think it's a CF2 one. Or the RCR2, CF2 anyways.
Yeah, and Cladosporium.
Yeah.
I don't remember. But a possibility also, because we tried very hard to find some targets of AVRM47 and AVRM3 and so on, but we didn't find any very interesting target, or we could not validate these targets. Perhaps it's not a protein. The target could be also something else than a protein. And in this case, it's a bit more complicated to find the targets. Okay, so I think I will have to move on. So I will continue with the last part of my talk regarding the regulation of effector gene expression. So on that part, that's a part of the project that I developed with Jessica Soyer, also researcher in my group. We look at different levels or elements of regulation, the role of the chromatin structure, and particularly of histone methylation, and of transcription factor on the regulation of effector gene expression. For the moment, we have not looked yet at some environmental or plant signals that could be involved. So I will go back to the structure of the genome of Leptosphaeria maculans. As I previously mentioned, this genome is a two-speed genome structure, with gene-rich regions that contain 95% of the genes, and repeat-rich regions which only contain 5% of the gene but represent 1/3 of the genome. These regions are invaded by transposable elements that were truncated and inactivated. And in these repeat-rich regions, these regions are enriched in biotrophic effector genes, which are expressed at all the biotrophic stages of infection, and they are not or lowly expressed during in vitro growth. While the gene-rich regions are enriched in effectors, which are expressed at the late stages of infection in petal and stems. So we have different waves of effector gene expression with contrasted genomic locations. So we investigated the involvement of the chromatin structure on their expression. So first, I would like to remind you that the chromatin can adopt two forms: the euchromatin, which is the part of the chromatin which is open and in which the genes are expressed. It's often gene-rich, and one characteristic modification in this region is the remethylation of the lysine 4 of histone H3. And heterochromatin, which is the more condensed form of the chromatin. It's often repeat-rich. The genes which are located in these regions are silenced, and there are two characteristic marks: the methylation of the lysine 9 and of the lysine 27 of histone H3. So what we did, we targeted this characteristic modification, and using chromatin immunoprecipitation and sequencing, we were able to localize these different marks all along the genome of Leptosphaeria maculans during in vitro growth. So what we found is that the gene-rich regions are enriched in euchromatin modification, but also in one heterochromatin modification, the H3K27, which is characteristic of facultative heterochromatin, while the repeat-rich regions are enriched in constitutive heterochromatin, the H3K9 methylation. But the biotrophic effector genes are also enriched in this heterochromatin mark, and including the avirulence effector genes. So what we did already 10 years ago was to inactivate or to silence the expression of methyltransferase KMT1, which is responsible for the deposition of this H3K9 methylation. And what happened when we inactivated KMT1? We could see a high increase of expression of the gene that were located in the middle of transposable elements. 41% of the effector genes that were located in this region were upregulated. However, the level of expression they had was not so high at the same level, as during infection. So at that moment, our working hypothesis was that during axenic culture, the effector genes, which are located in the middle of transposable elements, are located in heterochromatin regions. Their expression is repressed by the presence of H3K9 and controlled by KMT1. And at the onset of plant infection, the chromatin structure is open by a mechanism we don't know yet. And then the promoters of the effectors are accessible to specific transcription factor. So we decided to look at the transcription factor that could be involved in this control. So our hypothesis is that we have a two-layer regulatory mechanism: chromatin control and transcriptional control. So we look at different transcription factors that were described as involved in the control of effector gene expression. And one of them was a transcription factor which has been described in dotidomycetes, in several dotidomycetes, as involved in the pathogenicity and in the control of the expression of secreted proteins or effectors. It's called PF2. It was first described in Alternaria brassicicola as a virulence factor and as involved in the regulation of several secreted proteins. It was also studied in Parthenospora nodorum and Pyrenophora triticirripensis, with again a role in virulence and in the regulation of tox effectors. And an homologue had been also identified in Leptosphaeria maculans. So we decided to study this homologue and to see if it was able to control the expression of some of the Leptosphaeria maculans effector genes. So when we look at the expression of LMPF2 here in red during cotyledon special and stem infection, you can see also with different bright colors the expression of the different avirulence gene of Leptosphaeria maculans, and in black the expression of KMT1. So you see that LMPF2 almost follow the same expression pattern as the avirulence genes, while KMT1 seems to be regulated at an opposite manner. So that's already something interesting, and we wanted to go thorough with LMPF2. So Colin Clairet, during his PhD, generated different fungal material to study the role of PF2 in the regulation of effector genes, and his work was recently published. So he first inactivated KMT1, inactivated PF2, but he also overexpressed PF2 in a wild type context and in a mutant, KMT1 mutant context. So what we obtained when we characterized the corresponding transformants, so we could see that the transformants were growing normally. When we overexpressed PF2, we did not have conidation anymore, or low level of conidation. We did pathogenicity assay. We found that the inactivation of KMT1 decreased the pathogenicity of Leptosphaeria maculans, while the inactivation of PF2 abolished this pathogenicity. And for the overexpression, we did not have enough conidia to test pathogenicity using that strategy, but we could inoculate with mycelium plug. And in this case, at least for 3 of the transformants we tested, we had pathogenicity. Then we decided to perform RNA-seq analysis during in vitro growth to see what was the effect of this inactivation and overexpression on the expression of effector gene. So we looked, there were not so many genes that were up or down regulated, but there was a major effect of the overexpression of PF2 in a KMT1 context. And here I will not comment on all the results. You can have more details in the publication, but I just wanted to mention a few results. So there is a major effect on effector genes. So when we inactivated KMT1, but also when we overexpress PF2. And for the genes that were located in that were enriched in the mark H3K9, we could see an effect on induction, a high induction of expression when we inactivated KMT1 and overexpressed PF2. And among with this gene, we have 8 out of the 10 avirulence gene present in the strain we use for this study, and 20 out of the 36 effectors which are associated with H3K9 were upregulated. So to conclude on this part, our hypothesis was that there was a 2-layer regulatory mechanism involving a chromatin remodeler and specific transcription factor, and we could, indeed validate this hypothesis, at least for part of the effectors of Leptosphaeria maculans. But there are still a lot of open questions. We don't know which are the direct targets of PF2. What is the signal or the signals, environmental or plant signals involved in chromatin decondensation at the onset of infection? What are the actors, the chromatin remodelers involved in the decondensation? And finally, do all the region are decondensed at the onset of infection, or do we have some different waves of condensation and decondensation all along the infection cycle? And that's not an easy question to answer because it's quite difficult to look at the chromatin structure during plant infection. So to finish, and before taking question, I wanted to thanks all the people from my group at the Biogère Institute, the people who are still working in the team, and the PhD, postdoc, and master student who worked on the project, the different funding we had on these projects. And also wanted to mention our collaborators on different topics: structural biology, oilseed rape breeding, plant and fungal epigenetics, and effector biology. And thank you for your attention. And I'm taking your questions, if you have some on this last part or on other parts of my talk.
Thank you very much, Isabelle. Very nice talk. Any question? Oh yeah, I see. No, I see at least hands clapping. So I do have a small question that might be more like on the application side, because I'm always interested in that side. So you said at the very early stage of your presentation, I think, that you are collaborating with eventually industrial partners on this topic. And so I grasped that you had on the resistant protein, the RLM proteins are spread in the field, right? That's what you said, and that's been a long time already. Is that correct?
Yeah, It was for the first resistances that were used, knowing that there was a resistance in the cultivar, because some were used before, but people didn't know that there were some specific resistances at that stage, at that moment. Yeah.
So they are very resistant. They're like very stable proteins then, I suppose. And how do you, just the packaging, I was thinking, how does that work? Like you isolate the protein and then you have a certain treatment of it and then you spread it in the field?
No. Well, no, we do not use, we do not produce the proteins. They are present in the cultivars. We use, the breeders are choosing some rapeseed which are already resistant, and they crop them with some susceptible ones, but which have other great characteristic for them, that grow very well, or
I see. So it's a
produce
selection.
good oil. They select them for this resistance. So they introgress the resistance. They can introgress resistances from other rapeseed or from other Brassica species also. From Brassica rapa, Brassica oleracea. So one of our role, interaction with breeders is also to identify new resistance sources by looking at genotypes, many different rapeseed genotypes, but also other Brassica species, and trying to find way to identify new resistance sources, and to be able to predict the durability of these resistant sources, and to give some advices on the way to combine or alternate these resistances also.
Yeah, thank you. I see Minuka has a question.
Yeah, hi, that was a great talk. And I just had a question about the kind of methodologies you use to present some of your results. So I noticed that you had RNA-seq at the end. So I'm just curious what kind of methodologies you use in your day-to-day work in your lab, like yeast to hybrid, maybe transformations.
Well, sorry. We mainly work on the pathogen side, and that's right that we are not in our lab. We do not transform rapeseed, for example. So that's a limit we have. But we work when we want to test some interaction. We work on Nicotiana bentamiana using transient expression with agrobacterium. So that's a way for us to look at the compartment, the plant compartments targeted by effectors, or to validate some plant targets for the plant targets, we used these 2 hybrid, co-immunoprecipitation, or Fred Flim in collaboration with some colleagues in Toulouse. So we have different ways to validate. And yes, we do some RNA-seq analysis, genomic, comparative genomics. We predict also the structures now with the 3D structure using AlphaFold. Before we still try to produce some proteins in heterologous system using Escherichia coli, E. coli, or Pichia pastoris. And we have some nice collaboration with people doing some crystal structures or NMR. So many different aspects. Yeah.
Great. Thank you so much. Appreciate it.
Carol, you may go.
Hi, thank you very much for a nice talk. I have a question. Did you check the actual methylation changes during infection in the plant, or in the fungus during infection in the plant?
Well, it's quite difficult to look at, or at least what we can do is some cheap qPCR in order to look at the changes in the level of histone methylation, but we can only do that by targeting a few genes. And we tried very hard to be able to look at the whole genome methylation, histone methylation, but it's very difficult to discriminate the pathogen from the plant. We would need to be able to to sort the pathogen and the plant, because if you try to sequence directly the infected plants, I mean the modification, the histone modification we target are present in the plant and in the pathogen, so it's not a good way to discriminate. And the plant genome is really much bigger than the fungal genome. So if we sequence, it will be very difficult to find the fungal signal.
What if you pull down with, like, for example, your target, right? For chromatin immunoprecipitation, you use the target to pull down the DNA, and then you check the modification marks.
Yes.
Because I think that is very interesting to know if the change is actually okay, what kind of change is okay during the infection process.
Yeah, that's really something we want to know. But yeah, as I said, for the moment, we can just look at a few genes, like effector gene we know are enriched in H3K9 or H3K27. So we can target them, amplify them by qPCR, and see if they are still in heterochromatin region, or if there are some euchromatin marks during infection. But yes, we would like to know more what happens at the
I have another question. Like, for example, also, like, there's also H3K36, which is also an activation mark. Did you by any chance look at other modifications and how they regulate expression in maculans?
No, not yet. For the moment, we really focused on H3K9, H3K27, and no other ones. We focused more on trying to find what were what happens during infection if we had some remodeling, some histone demethylation that could suppress, eliminate these marks. But for the moment, we have not found which one was really involved. So we generated many different mutants, and we need to determine which histone demethylase is responsible for which modification.
All right.
But we didn't try over.
All right. Okay. Thank you.
Bradley.
Hi, thanks again for a great talk. This is a relatively specific question, I guess, and I apologize if I missed it. Do you know the size of the intergenic region between AVRLM 10A and B? And is that size similar for other? I also wanted to.
It's quite huge compared to the others. It was like more than 7 kb.
Oh, okay.
So yeah, at the moment when we try to published the story about AVRM 10A and 10B. There were many questions from the reviewers about the role of this intergenic region. So we did quite a lot of experiments trying to complement with or without this intergenic region. We know that there are no transcripts in this region. There are only transposable elements which are inactivated. But for sure, if we do not complement with the whole region, expression of the gene is not very high. So it's important for the regulation, but there is no additional genes in this region. But yes, for the other copies we found in the genome of Leptosphaeria maculans, the intergenic region is really shorter, like less than 1 kb.
Very interesting. Thank you.
So
All right. Yeah.
Go
Marielle.
ahead. Well, I was just wondering, since there was a lot of question and I'm new to it, so should we do the meeting with the fellows, or are the fellows all right with what they heard right now? Maybe Minuka and Bradley, I think you were the host for the next part, so feel free to.
I may interrupt here, but maybe I suggest that you give it a try for a few minutes. Maybe there are some people that prefer to be, you know, only with students. So it gives a chance to ask a few more questions, and then it doesn't have to be long, right? So I would still try.
Okay, good. So do we stop for just a few minutes so my Prof Fudal can take a little breather, and then we meet in, like, I don't know, with 12:08? Is that all right?
Yep, sure.
Okay.
Okay.
Thank you, everyone.
Thank you. Thank you very much, Isabelle.
Thank you, and hope to see you soon, even if it's not in Asiloma.
No, probably, yeah.
Yeah. Perhaps
Sometimes
in Dublin next year.
that would be great. Let's aim for that.
Yeah.
Okay, thank you again. And I will quit from here, so and let you in the good hands with the students. Goodbye.
Bye, Isabelle.
So we'll be
Bye,
back
Marielle.
in 4 minutes. Okay.
Yeah. Okay, see you in a couple of
Okay.
minutes.
Okay,
Thanks.
great.
So does anyone else have any questions for the Q&A session?
Yeah, I've got just a kind of a more general question. You started at, what was it, Biogure in
Yeah.
2014. I was just curious more about the center and, you know, the facilities that you have there and how you enjoy working there and, you know, what it's like being in that part of France and that kind of stuff. So maybe you could elaborate, enlighten us on what it's like working there.
Well, so I'm working for INRAE. It's the National Institute of Agriculture, Food and Environment, and that's right. But usually the INRAE centers are located in the countryside with lots of fields outside in order to do some field experiments.
Right.
That's what it was until last year. Now we moved to a more a campus with lots of buildings and less fields. So it's a bit more complicated now to do some field experiments, even if
More like
we
a campus
have
now.
some in Versailles, so it's not so close. So it needs some more organization and some people who are dedicated to doing the field experiments and looking at the symptoms and tell us you can go come and collect some plants, or it's time to do some experiments and so on. But and then while we have, what is interesting, at least in France, in the French system, we have many permanent people, and usually it's not a work with one PI and his PhD students, postdocs, and so on, but you have several researchers in each team, and we are working together, trying to share the project we have, and there are also some permanent technicians. So that's something good, because you can do experiments during a long time with the same people who know quite well the model you are working on. So we have some people dedicated to molecular biology, to microbiology, cytology, some bioinformaticians to help us with the RNA-seq analysis, for example. But it's a bit more complicated to find money for the project in France. Yeah, so you have permanent people, your salary is, you're paid already, that's great. You do not have to find money for you and your colleagues, but we have, it's more difficult to find funding for the experiments.
Yeah, so is it maybe similar to the system in Canada where a lot of it is through grants and different granting agencies, or, you know, it's interest groups or something like that? Is that similar the way the funding is done there?
Yeah, we have some money gave by directly by the French government, but it pays mainly the growth chamber, a few things like this, electricity and so on. But then we have to find money for the rest. We have a national agency for some big project. At INRAE, we also have some, we depend from the plant health and environment division, but also can pay for some small project to start new, well, to help the new idea we have, to support new ideas. And then we have some first results we can apply for bigger grants. There is also European project, the possibility to have European project, but it's a bit more complicated. And you can have also project with private companies or PhD paid by private companies, depending on the project you have. So for us, it's more about breeding, about identification of new resistant sources. For other person in the institute, it's on fungicide or new biocontrol molecules and their mode of action. The ability of the pathogen to develop resistance mechanism against these molecules. Usually we try to combine all these sources of fundings, and in our group we are also paid by some different breeders and institutes in order to characterize their new plant material. So when they develop new resistances, they ask us to determine what is present in the material they have.
Yeah, very cool. Sounds like a very collaborative environment. Requires a lot of communication, but it sounds like it's worth it. The work is fascinating.
Yeah, that's nice, because we are not alone on our project, and we can discuss with the colleague, have some advices, and yeah. And
Excellent.
we do not teach so much, at least in RAE, we are only doing research. We can do some teaching if we want, but we do
Separate
not
from
have
universities
so many
or
hours.
yeah. Okay.
I understand this may not be your personal area of expertise, but since you brought up the idea of the Institute doing some work with small molecules, I'm very curious, what kinds of small molecules do you guys work with and work on? Because I know some people are trying to develop new fungicides, like things that will kill the organism. I've heard some really fantastic stories about small molecules that have more to do with detoxifying or preventing an organism from producing a toxin. I'm just curious if you could talk in broad strokes about what kind of biology you're looking to target with these molecules.
And where they come from.
It's not my main project, I would say. So it's more other
Very
people
fair,
who are
yeah.
working on that. So and even on effectors, I'm mainly focusing on protein effectors, but other people in Biogen are working on secondary metabolites or small RNA. But on the fungicide side, I mean, the molecules are more, for the moment, mainly classical chemicals and also some molecules produced by bacteria and so on, but probably more secondary metabolites and no particular
Very
new
cool.
molecules. That's all the questions. I don't. Are you all in the same region of Canada or in many different institutes and universities?
No, I think that we're from all around. So I know that myself and Chris, at the very least, we're in Ontario.
Inuit
But
as
other
well.
folks can chime in. Other folks can chime in. I know that we have some people from, say, British Columbia. We have some throughout. So kind of pan-Canadian representation.
Okay.